Please welcome the President of the American Society of Nephrology, Dr. Prabir Roy Chaudhry.
Thank you all, and good morning. Over the last few days, I've been incredibly excited and energized by the amazing vision and science and commitment to kidney care that has been the hallmark of Kidney Week 2025. The promise of the future, however, is always grounded in the contributions and commitment to nephrology of all the friends and colleagues who we sadly lost this year. We clearly stand on the shoulders of giants. Thank you for celebrating the legacy of care, hope, and discovery that they left us as we continue to strive for a world without kidney diseases. Today's plenary session is dedicated to the significant and exciting advances in xenotransplantation that combine genetic engineering and immunology. These advances could provide both a solution to the organ shortage and also hope to the thousands of patients with end-stage organ failure. Building of Dr. Perkovich's presentation on Thursday, yet another way to hopefully be able to cure kidney diseases. 2024 marked the first genetically modified pig-to-human kidney transplant, and earlier this year, the Food and Drug Administration announced clearance of clinical trials for genetically modified pig kidney transplants in humans. ASN is delighted to have been able to bring together an incredible expert panel of pioneers and leaders in xenotransplantation, including David Cooper, Robert Montgomery, Leo Riella, the three of them being led by our moderator, Dr. Paul Klotman. I know that we all look forward to hearing the insights of this panel on the many different aspects of xenotransplantation, going all the way from genetics and immunology to surgical protocols, ethical issues, and payment. And with that, it is my true and very distinct pleasure to hand over this session to our moderator, Dr. Paul Klotman, the current president of Baylor University, and most importantly, Paul, a card-carrying nephrologist and good friend and colleague. Paul? Come on in.
Well, welcome everybody. Is it on? Can you hear me? Hello, hello? Yes. Welcome to Houston. If this is your first trip to Houston, Texas, the weather is like this year-round. Yeah. So we're really excited this morning to have a terrific discussion on one of the really cutting-edge fields. And our guests are Dr. David Cooper, who trained in the UK as a cardiothoracic surgeon in Cambridge and London. In the early 80s, he worked with Christian Barnard, the pioneering heart transplant surgeon from the University of Cape Town in South Africa. In 1987, he relocated to the USA and is currently at the Mass General. And his major research focus is in xenotransplantation with the aim of using gene-edited pigs as sources of organs, cells, and corneas for transplants. He's a member of the medical team that carried out the very first pig transplant in a patient in 2024. So fantastic. Leonardo Riella is the Harold and Ellen Dancer Endowed Chair in Transplant Surgery at Harvard Medical School and the Medical Director of Kidney Transplant at the Mass General. And I just learned that your father was here 51 times. That shows bad judgment, but that's okay. I was looking at that board. I couldn't even reach down to hit the 50. Anyway, that's another story. He completed his internal medicine residency at the Brigham in nephrology and transplant fellowship in the combined Brigham and Mass General program. His research is focused on understanding the mechanisms of immune regulation on the development of novel approaches to promote organ tolerance. His lab is tackling some of the immune barriers to successful xenotransplantation. And Dr. Montgomery is the chairman of surgery and the director of the NYU Langone Transplant Institute in NYU. He received his MD from the University of Rochester School of Medicine and did his surgical training in transplantation and completed a fellowship in human molecular genetics at Hopkins. And for over a decade, he has served as the director of the Comprehensive Transplant Center. Dr. Montgomery is part of the team that developed the laparoscopic approach for living kidney donation, and in 2021 led the team that performed the first gene-edited pig to human xenotransplant, and has performed 8 pig heart transplants so far, and has the coolest boots ever. I mean, ever. I love those.
When in Texas.
When in Texas, where you got to wear boots. You got to. Anyway, so this is an important topic because The Economist just had an article, I don't know if you saw that, last week on how pig transplants are going to solve everything. And, this has been a topic that has been viewed as a potential answer to the challenges of organ procurement. But this year has marked a significant milestone in that first living human received a genetically modified pig kidney transplant. And the FDA has now approved clinical trials. So this is a huge step forward. And I would like to ask each of you from your perspectives, so why now? So Dr. Cooper, maybe you have the longest perspective on the preclinical work. Why is it all coming to fruition this year?
40 years ago, when you put a pig organ into a baboon, for example, you got hyperacute rejection, rather like when you get an ABO incompatible graft. But we've been able to work over the last 40 years to improve the situation in 2 respects. One is we've been able to genetically engineer the pig to protect it from that human immune response. And this is very important for some of you who may not have actually thought about this. This is the first time in transplantation that we can actually modify the donor as opposed to modify the recipient or treat the recipient. And we've done that in 2 things. One is by genetically modifying the pig, we've knocked out the antigens against which humans have antibodies. We have antibodies because we develop them when we have our gut colonized, because some of the viruses and bacteria have the same sugars on the surface as the pig does. So we become sensitized to the pig, and that's why we have hyperacute rejection when you put a pig organ in. But secondly, we've been able to utilize these novel immunosuppressive agents that have become available in the last 25 years. And we found that they are much more efficient at protecting the recipient from developing new antibodies against the pig. And so that those 2 things, the genetically engineered pig and the novel immunosuppressions, they've made a huge difference to the progress we've been able to make. So that's why I think we're onto something now that we will pursue, and I think will revolutionize transplantation. And I think the drugs that I've been mentioning will revolutionize allotransplantation as well. My colleagues at MGH, Dr. Tetsuo Kawai and Robin Pearson, have shown in the monkey model of an allotransplant that if you give this drug a loading dose up front, but then you give it once a week or even once every 2 weeks by injection, no other drugs at all, no induction therapy, no other maintenance therapy, you do not get rejection. It's quite remarkable to me. Now, with an xenotransplant, you do need some induction therapy in order to make sure that you don't reject, because the response is much harder. But I think we're on a verge of revolutionizing transplantation.
Dr. Riella, what are your thoughts about that?
Yeah, so it has been incredible. What we've learned goes back, I think, to the basics, to the biology, trying to understand exactly what were the incompatibilities, and then finally having tools to be able to modify that and improve that. And when we think about 3 basic buckets, we think about removing red flags from the transplanted kidney coming from the pig, in which we recognize, as Dr. Cooper was saying, with natural antibodies, neutralizing infections that could potentially transmit, and third of all is how can we improve some of the compatibilities in the complement system, in the coagulation system, since we separated from pigs more than 80 million years ago. And CRISPR-Cas was a gene editing tool that basically changed the game in xenotransplantation, and what brought us now to where we currently are in is all the preclinical work, all these animal models in which pig kidneys were surviving more than 2 years in monkeys. And so I think we had the evidence that kidneys were sustaining life, The question is, how do we then move to the clinic and start testing in humans? Because of course it's going to be different, and it's going to be a learning curve.
Dr. Montgomery, what do you think?
Well, I have a slightly different perspective. All the work, all the preclinical work that was done was very important, but we can all think of many examples of things that have worked well in animals and then have not translated into humans. And, I spent 20 years basically studying sensitized patients and doing ABO incompatible patients. So a very similar primed immune system, preformed antibody, preformed T cells that, we had to work out a lot of different therapeutics and approaches to how to reverse rejection, how to take care of these patients. So the big jump, that I felt that was made by us and others was to find a way to get these into humans, because we were pretty confident that once we got one of these organs into a human, we would have the tools we had accumulated, the approaches to be able to take care of these really complex immune systems. And so it was the decedent model, doing these initial transplants in a risk-free environment in brain-dead recipients who had wanted to donate organs, but for whatever reason their organs weren't suitable, and so we asked the family to donate the body to heart beating in an ICU to test these organs. And I think that got the FDA in a different mindset when it came to presenting single patients to them for
that's pretty remarkable when you think about it, that the decedent patients actually were a big part of the transition. What do you think now are the major barriers? I mean, we have an FDA-approved trial going. There's still a lot of barriers to success. So from your perspective, and I'll start with you this time.
Sure. Yeah, so the good news is I think both groups have shown that as far as the physiology of the kidney, a pig kidney can do almost everything that a human kidney can do. And the things that it doesn't, we have redundancies, that cover for that, and we also have drugs that we can use. So it's really all immunology now, which I think we all would say is wonderful, because, we're basically going through what, 70 years ago, the early, pioneers in allogeneic transplantation were doing, figuring out what's the right immunosuppression, but this is going to happen in a much more accelerated fashion. And then, as David said, we also have this great advantage of being able to manipulate the organ itself, which we never had in allo transplant.
Yeah. So what are your thoughts about the big barriers yet?
No, I agree with Bob. I think the immunological challenge is going to be, I think, the one we're going to be dealing with going forward. What we are now is in a different environment when we think about the research that we can do. And so I think COVID was one example of a research pattern in which we try to get results very quickly and apply back into what we are doing. And I think Xeno, we are doing the same thing. So, we are running assays in which usually may take months to do the analysis, go back and finalize it. And now we're doing a very quick turnaround and learning with each case. And every case is different, but they add on into supporting what we are doing and potentially adding those modifications that we believe are going to be important to sustain the kidney in long term.
Good. And Dr. Cooper, what do you think are some of the barriers left?
Well, I think the main barrier, unfortunately, is that although we don't see rejection anymore in these pig kidneys, which is remarkable, I think, having seen rejection for 30, 40 years, we don't see it anymore. But we do see thrombotic microangiopathy, and it's associated with proteinuria. And the proteinuria can be very severe. In the baboon model, for example, we can lose so much protein that we also start to lose the anti-CD154 monoclonal antibody, which is the key immunosuppressive drug, which is a protein. We lose that in the urine as well. And if you don't keep an eye on that, you will get obviously such a low level that you then will get rejection. So we still have this major problem, and I think it's an immunological problem, because if we give the same immunosuppressive therapy, we don't see it in a heart transplant model, of course. So it's something to do with immunological. And I think we will have to have a better pig, even though we have pigs now with 10 genetic manipulations. I think we need a better pig, or we need some way of preventing this proteinuria from developing, maybe by adding, say, an anti-CD38 monoclonal antibody to the regimen. So we have a problem which we didn't expect to see, and it's quite a major problem.
So I like the concept of a better pig. We all need a better pig. That's true of barbecue as well. Mm-hmm. So, I've spent much of my career working on HIV, and endogenous retroviruses are a big problem. Porcine endogenous retroviruses exist. The safety concerns are not inconsequential. Really interested in how you think about how do we deal with the PERVs that are there, and how do you monitor? What are your thoughts?
Well, look, this almost killed our field. We all remember in the late 90s, the porcine endogenous retrovirus story really had a chilling effect on xenotransplantation. Yeah. And luckily, people like David, were able to push through that, or we wouldn't be here having this conversation.
Exactly.
So I think you have to have a strategy. Right now we're working with United Therapeutics. They're the sponsor of the trial that we're working on, and they have bred PERV-C out of their pigs, and it's the AC recombination that people worry about. So that's been the strategy that we've had to reduce that risk. And we've also built very sophisticated platforms for looking at all the zoonotic pathogens of interest.
Yeah.
So eGenesis, which are the pigs that we have been using in our human studies, they had a slightly different approach. So they have used CRISPR-Cas to neutralize all the porcine endogenous retroviruses. So there's a total of 59 inactivations. The safety in terms of infection perspective of these pigs, I think it's much greater than if we think about a deceased donor. And because these pigs are grown in isolation, they're bred, in a very specific manner, they're testing prospectively, and not only for PCR or serologists, but there are all these metagenomic platforms that run assays to detect any possible xenopathogen.
Are they looking at reverse transcriptase? Are they?
Yes,
okay.
yes. And then
it's reassuring to know.
Yeah, and then it's not only the active monitoring, but also passive monitoring. So once you transplant these patients, for the first couple of months, they're tested weekly for any potential evidence. So overall, we think that the risk of transmitting a new infection is very low, but we got to be cautious. That's kind of regulatory-wise, I think, very wise.
And your thoughts about that, David?
No, I think the infection problem, my infectious colleagues, infectious disease physicians, think that the problem will be very similar to the allotransplant, that there will be opportunistic infections, but they don't see a big problem from pig infectious microorganisms in the human recipient. I think, coming back to the genetic engineered pig, I think there are ways we can perhaps solve the problem of proteinuria and microangiopathy. There are some suggested genetic manipulations which were suggested a long time ago by one of our colleagues in Boston, CD39 and CD73, which actually are anti-inflammatory, and much of this may be inflammation, say, of the podocytes and so on, and anticoagulation. And we need something, because the microangiopathy is probably a coagulation problem. So there are ways we can add to the 10 gene manipulations we've got now to improve them even better. And I think that's the sort of approach we probably should go to. I think doing it with drugs is going to be difficult, because you're going to suppress the patient more and more and more. And I think we need to still concentrate on the pig.
So what do you think of the timeline? I mean, we have a clinical trial going on now. It's taken 15, 20 years to get to this point. Where do you see the future in terms of, success? What do you think is going to happen? I'll start off with you, Dr. Montgomery.
Well, so think about it. There were 30, 40 years of preparation, but in 2021, we put the first gene-edited pig kidney into a decedent, and now 4 years later we're in clinical trials. Where has that happened? In something this complex.
Right.
So I think we're in this acceleration phase. We're learning a tremendous amount about the phenotypes. They're different than what we see in allotransplantation, the rejection phenotypes, the inflammation phenotypes. We now have AI. Once we can, garner enough information in terms of understanding these early phenotypes, again, this is an accelerator. And I think we're going to also be able to draw from the allo database to inform what's happening in xeno. So I see this as moving very rapidly, actually. But to your point, Leo, we're still learning. Like, it's sort of like repairing an airplane, in the air, right? We're still learning a tremendous amount from each case that we do. Mm-hmm. So fortunately, some of the trials that we're involved in are adaptive type of trials, where I think we're going to be able to make some changes as we do these trials. So.
And do they give you the flexibility to do that in the?
Well, so they're seamless safety into a phase 3, and there's going to be opportunities, in our trial after the first 6 patients, after the safety run-in,
right, to
make some changes at that point.
And what do you see as the FDA approval process when successful?
So the FDA, I'll have to say, has been so collaborative across the entire process. I've been involved with the FDA before, but I've never seen a group as the current one that has been working with us in Xeno.
So this is a shout out to government.
Yes,
it's a good thing.
Yes, okay. I think if we,
doesn't happen often, but it happened this morning, Sunday morning at the ASN.
Yes, no,
government's doing something right.
No, we're very proud of the work we've done with them, because we truly felt that as we were sharing the data, as we were proposing new strategies to keep advancing, there was this positive feedback loop where they were not trying to create barriers, but they were trying to see, okay, how can we make it safe? How can we improve it? Bringing up new suggestions. And truly, I think this is what we hope from our regulatory agencies.
Yeah.
And I'll add the piece about the patients, right? And yes. I think this is something that Bob also alluded many times. We are now talking about xenotransplantation and moving to the clinic really because our patients have had such a positive reaction to the first decedents that were done, the first living human transplant that were done. And if it was not for the patients advocating for us to advance xenotransplantation, we wouldn't be here. So I think a shout out, to all the patients and also the societies that put patients together for us to hear their voice.
Yeah, I see Vinita Kumar in the audience. She really organized this session with patients and the FDA. And I can tell you that changed everything.
Yeah.
So they listened to, patients who were on dialysis for years, and really were very responsive to that session. So the patient voice, I think, is more important now than it's ever been.
David, what are your senses of
Well, I'm a little concerned because this proteinuria problem is so marked in the baboon, and it is occurring in the patients as well, that if we don't solve that problem relatively soon, the FDA may put the brakes on this very rapid progress that we've been able to make. So I think it's a very important problem, and obviously we can solve these problems more if we've got good funding for the research. And this is something that we would need to do in the non-human primate model, because you need to follow for several months. So I think this is a critical time. We've got a new problem we haven't seen before that we now need to resolve. The other thing that we need to pay some attention on, which we haven't as yet, is that these facilities for breeding these pigs are very expensive to build and very expensive to run. And we won't have too many of them around the country or around the world. And I think we need to spend more time now on considering new forms of storage of the organ, that we can actually ship the organ internationally if necessary. And there are new projects coming along, new proposals for isochoric supercooling, which is a remarkable new approach, where you just take the organ, put it in a box and ship it off. And if we can do that, that will make facilitating extension of xenotransplantation globally and nationally much easier to do.
So you've alluded to some of the technical barriers. That's a technical barrier. Leo, what do you see as some of the other technical barriers for success?
Yeah, I think the scalability of this, is going to be very important. And if we think about, we have over 600,000 patients on dialysis. Imagine now if we can actually bypass the dialysis and place these patients with a pig kidney. We're not comparing now the xenotransplant with a human kidney transplant. I think we're not there yet, but we're definitely comparing with dialysis. I think our bench, is dialysis, and I think we can go better. I hope that we can do that we can do better than that. And I think the scalability means that if now we have 10% of our patients, 15% of our dialysis patients on the waiting list, and that's just a very small fraction of the patients who truly would benefit. So we have to think about, it will take, of course, time until we get there, but we've got to start thinking about scalability and potentially, overcoming some of those challenges.
Bob, what do you see as from your perspective?
Yeah, I think what Leo said is really important. And, these more complex gene edits that are out there right now that are being tested require cloning pigs, and that is not going to be scalable. So we've got to figure out how to breed or use less complex gene edits that are more easily bred, so that we can do that. So that's what we've been sort of looking at the minimalist approach and these really complex gene edits, and we'll see how that all turns out.
So we didn't talk about this before, but David, could you? People don't realize what it takes to have a gene-edited pig. So can one of you talk about just the process of getting a gene-edited pig? So it's not like these pigs are on a farm someplace.
Well, you take a pig cell, and it could be one of several different sorts of cells, and you genetically engineer the pig through the CRISPR-Cas9 technique. There were techniques before that, but they were less precise, they were longer term, and they were more expensive. So this has been a significant improvement. But you manipulate the pig, you put in a gene, for example, a complement regulatory protein, a human complement regulatory protein. Pigs have complement regulatory proteins, but they're not very good at protecting against human complement. So we put in human genes and knock out these antigens in the pig as well. The two are very, very valuable together. And then they put that cell into a surrogate pig, and they breed the piglets from that one, and the piglets express the transgene, or they knock out the thing. So it's a time-consuming thing, and every time they do it, it's another several months before you get those new pigs out. So it's going to take a time if we need to put in new pig genes, it's going to take some time to get those pigs, and then you've got to breed enough of them to test in the monkeys, and then enough to test in the patients, and so on. So it's going to delay us, unfortunately. We thought we had a pig that was ideal, and in fact we now find that it's not quite as ideal as we thought, although it's much better than the pigs we had in the past.
But it's gene editing 60 to 70 genes in some case, and starts off with a single cell.
Yeah, it's cloning, right?
It's like cloning the sheep.
It's Dolly.
Like cloning
Dolly sheep, yeah.
It's like Dolly. Yeah, but it's hard to believe. You're basically cloning the pig, and then you have to breed them. And there are a lot of different strategies. You want to talk about that?
Yeah, I think, the two important things, I think, is thinking about what strain of pig are we choosing from. And so there's this mini swines that go 75 to 90 kilos, and there's this, what we call the big whites, which can go up to 300 pounds. And so there has been different strategies about which pigs to use and how can we then get the kidney of the right size or the heart of the right size. So on the mini swine, which is the one we've used, is the Yucatan. They come from Mexico. They have 75 to 90 kilos, and we use those cells to generate. But then they're placed actually in the surrogate mother, which is a big white.
Right.
And the beauty about the pigs is that the gestation is 3.5 months, and by 1 year we can use the organ. And they give between 6 to 12 piglets. So the scalability, and that's why pigs were chosen in part to be donors.
You're not putting any tariffs on that. Yeah.
No, tariffs.
So we've talked a lot about different strategies to do this. there are some ethical concerns that people have raised over the years. It's probably good for the audience to hear what are some of those. Maybe Bob, you could start.
Yeah, I mean, I've had conversations with our friends whose careers have been really focused on animal rights, and they've been very productive conversations and discuss things like, could we remove one kidney from one of these pigs and have them survive? And, could we optimize the number of organs that we get from each pig? So I think very sort of positive, thoughtful approaches. How are the pigs being cared for? What does that look like? It looks totally different than the industrial raising of pigs, that provides, the 1.3 billion pigs a year that we consume on this planet. So, I think the reassuring the public and the folks who are really laser focused on this is a really important thing, that these pigs are being cared for in the best possible setting, stimulated, that we're thinking about how to minimize the number of pigs that we use. In xenotransplantation, all those things are going to be critically important for public acceptability.
David, some of the early stuff was in primates too. So
if I could just make a point, we eat 100 million pigs a year in this country. So we're talking of a minimal number if we're talking about them as donors for organs. So I don't think we've got to put this in perspective. More than 100 million are killed each year in this country. The second thing is that Sir Peter Medawar, considered the father of transplant immunology, he said 50 years ago, he thinks people will go for the allo transplantation at the time, and now the same for the xeno, because they'd rather be alive than dead. And that puts it at a baseline, I think, that we have to sort of consider all the time. The last point I'd make, too, is that half of the patients, my understanding that half of the patients waiting for a kidney transplant in this country are diabetic. And we have the opportunity now of curing their diabetes as well. Back in 2009, we had the first monkey that was diabetic that was cured of its diabetes for a year. We followed up for a year, didn't need any insulin for the whole year because we put in pig islets into the monkey. And I think here we have an opportunity of treating the renal failure and at the same time treating the diabetes if we put in islets with the kidney. Now, if we can't put the kidney in because it's all getting proteinuria at the present time, I believe we should be putting in pig islets into every patient who's having a human organ transplant, a human kidney transplant, because they've got to have immunosuppression anyway, so you might as well cure their diabetes at the same time as curing their renal failure.
Leo, there's some really interesting work about potentially doing a co-transplant with a thymus and trying to educate T cells to be tolerant. What do you think of that approach, and do you think there's a future for that?
Yeah, I think we do have to think about how can we best overcome the immunological barrier. Bob and the Columbia team who started with the process about transplanting the thymus with the kidney with the idea of there will be some re-education of those T cells have been really interesting. I think we have to test that hypothesis. So tolerance at the end requires us to stop immune suppression and see the absence of an immune response against the transplanted organs. That ultimately, there's no blood test, there's no assay that can prove tolerance until you remove the immune suppression and there's no rejection, and the immune system's still capable of fighting infections. So that's, of course, the holy grail. And I think meanwhile, until we fight to get there, I think we do have to come up with ways of minimizing the amount of immune suppressive drugs, since right now the cocktail it's a hefty cocktail that we start with. But we won't keep that hefty cocktail for life. I think the idea is what are the things that we can start reducing over time in order for us to get that right balance of infection and
you've been
rejection.
A pioneer in this, so
well, yeah. I mean, I've been using the organs, and it really is David Sachs and Megan Sykes who developed this whole field in large animal models and in the humanized mouse. And it looks very promising. It's amazing to think that you could that you could co-transplant a thymus from the donor pig and actually, affect the T cell repertoire, that human thymocytes could, be able to get signals and be deleted in the context of a pig thymus. It's an extraordinary idea, but it has worked in these animal models, and we have an N of 1, and we didn't reduce the immunosuppression, but we have MLRs that showed that that patient wasn't responding to the donor pig, but was responding to a third-party pig and a third-party human. It's crazy, but I think it's going to be important. It may not be. I'm not thinking about swinging for the fences and saying we're going to remove immunosuppression, but maybe we could reduce immunosuppression to lower levels.
It's a great idea. It's a really great idea. The other thing I've wondered, in the murine models, it's been very easy to humanize the mouse. From your perspective, what do you think is, how much of the human genome are we going to be putting in to these engineered pigs? David, what do you think?
I think we need to look at the immediate problems, such as proteinuria and so on, and not be too far forward. I worked with David Sachs for 9 years on the tolerance thing, and it's a very difficult problem to overcome. But I think we should not underestimate the potential of xenotransplantation. We think of the kidney transplants, heart transplants, and so on. But, for example, there was a group in Italy many years ago now that was curing Parkinson's disease in monkeys with Parkinson's disease by putting in pig dopamine-producing cells into the brains of the monkeys. And this was quite remarkable. They had videos of these animals much more mobile and so on after the transplant. And it was only stopped because the Italian government banned studies in non-human primates. But there are several studies like that in the past that I think have tremendous potential for xenotransplantation, and that we need to begin to investigate those again now that we have much better pigs and much better immunosuppressive therapy.
Well, we're coming to the end of the session, and I want all the audience to realize this is really a transformative time. And I don't think there's any question that xenotransplantation is here, and it's going to be successful. And I hope we have a chance to solve the problem with organ donation. I want to thank each of you, Dr. Montgomery, Dr. Riella, Dr. Cooper. You guys are leading the field, and it's been a wonderful, wonderful conversation. So, thank you very much, and that's the end. Thank you.
Thank you.
Thank you, guys.
Thank you.
Really
great. Well
really
done.
well. Really nice. Great work.